How does liquid-based cytology work for cervical screening? Imagine a process that makes detecting early signs of cervical cancer more accurate and reliable. That's what liquid-based cytology does. Instead of smearing cells directly onto a slide like in traditional pap smears, it collects cells in a special liquid preservative. During the screening, a healthcare provider gently brushes or swabs the cervix to gather cells. This sample is immediately placed into a vial filled with alcohol-based fluid. This step is crucial because it preserves the cells in their natural state and prevents them from drying out or getting damaged. Once the sample reaches the laboratory, it undergoes a series of processing steps. The lab technicians use techniques like vortexing and filtering to remove mucus, blood, and other debris that could obscure the cells. They then use a process called density gradient centrifugation to separate the cervical cells from unwanted material. The purified cells are then resuspended in a solution and carefully spread onto a glass slide in a thin, even layer. This thin layer preparation is important because it minimizes overlapping cells and makes it easier to see cellular details clearly under the microscope. The slides are stained using the Papanicolao method, which highlights different parts of the cells, making abnormal or precancerous changes easier to spot. Skilled cytotechnologists or pathologists examine these slides, either manually or with automated systems, looking for abnormal cells that might indicate early signs of cervical intraepithelial neoplasia or cancer. Compared to older methods, liquid-based cytology offers several benefits. It produces cleaner, higher-quality samples with fewer blood or mucus cells blocking the view. The slides are more uniform, which helps in accurate interpretation. Plus, because the sample is stored in liquid, it allows for additional testing, such as checking for human papillomavirus DNA, which is linked to most cervical cancers. This flexibility can improve overall screening accuracy and efficiency. Research shows that liquid-based cytology is at least as sensitive as traditional pap smears in detecting high-grade lesions, with some studies suggesting it may find more cases early. This means it can help catch abnormal changes sooner, giving women a better chance for early treatment. It also reduces the chance of false negatives and improves the consistency of results across different labs. In the bigger picture, liquid-based cytology plays a key role in cervical cancer prevention programs. By providing clearer, more reliable results, it helps identify women who need further follow-up or treatment before cancer develops. This approach aligns with global efforts to reduce cervical cancer rates through early detection and intervention. In summary, liquid-based cytology works by collecting cervical cells into a preservative liquid, processing and preparing them into a clean, thin slide, and then examining them carefully for abnormal changes. This method enhances the accuracy of cervical screening, making it a valuable tool in protecting women's reproductive health. Always remember to consult healthcare professionals for personalized advice and screening recommendations.